Welcome to this video about probability and Mendelian genetics. Now, everyone has most likely seen or used a Punnett square before. These are a very handy tool for depicting and predicting the outcome of a genetic cross. Let's start out with a basic one-factor cross in which both parents are heterozygous for gene A. In this scenario, the allele big A is dominant to little a. Since we only have one gene, the allele combinations are simple, and the potential offspring are shown in the box in blue. First, let's look at the probability of these results. Probability can be defined as the number of times a particular event or outcome occurs out of the total number of outcomes, and it can be expressed as a fraction, a decimal, percentage, or ratio. Based on our diagram of all possible outcomes in the Punnett square, the probability of receiving two big A alleles is 1/4. For heterozygous combination, it is 2/4, or 1/2, and for two little a alleles, it is 1/4. This can also be expressed by the ratio 1 to 2 to 1. Now for the phenotype. The dominant phenotype is expressed with both big A and big A little a genotypes, which I'll combine as big A blank. Therefore, the probability associated with the dominant phenotype is 3/4. For the recessive phenotype, the probability is 1/4. This can also be expressed as a 3-to-1 ratio. The Punnett square is still manageable for a two-factor cross. We'll use heterozygotes again. Here, the number of columns and rows we'll need is equal to the number of allele combinations that are possible based on the parent's alleles. For heterozygotes, this is equal to 2 to the number of genes, so 2 squared equals 4. I'll draw a 4x4 table and add the paternal and maternal allele combinations. The offspring are going to have many more possible allele combinations. And here are the 9 possible genotypes from this cross. There is only a single square of big A, big B, so it has a probability of 1/16. But for big A, big B, little b, there are 2 squares, giving it a probability of 2/16, which I will abbreviate as 2. As we go down the line, the probabilities are 1, then 2, then 4 for the double heterozygote, then 2, then 1, then 2, and then 1 again. Quite the numerical ambigram. But there are only 4 possible phenotypes from this cross. Let's go in reverse order and use the genotype counts we already have. The double recessive phenotype is only caused by a single genotype, so it has the same probability, 1/16. For recessive A and dominant B, the probability is 3/16 because we add both genotypes with little a. For dominant A and recessive B, it is also 3/16. And for dominant A and dominant B, we can add all of the remaining genotypes together, which yields 9/16. These probabilities can be expressed with the ratio 9 to 3 to 3 to 1. Now let's look at a 3-factor heterozygous cross. Here, we would have 2 to the 3 allele combinations, or 8 allele combinations. I'll construct an 8x8 table and add the possible paternal and maternal allele combinations along the side. And now to start adding the results. But I'm just kidding, this is way too much work. Luckily, other methods can be used to calculate and diagram Mendelian probabilities. First, let's look at the branch method, which combines simplified diagrams with probability calculations. We'll calculate phenotypic probabilities for a 2-factor cross. We'll consider each gene separately and assume that they are not linked. For gene A, we have the dominant or recessive phenotypes, which we know have a probability of 3/4 and 1/4, respectively. For gene B, we draw a branch with dominant and recessive phenotypes coming off of dominant A. But we also draw the same branch coming off of recessive A. In this instance, the probabilities for A and B are the same. Now, we can use the product rule to calculate the total probability associated with any particular branch. The product rule states that the probability that 2 or more independent outcomes will occur is equal to the product of their individual probabilities. The product rule applies to genes that obey Mendel's law of independent assortment. It is also applicable to other independent events, such as the birth of offspring or rolls of dice. The product rule can be expressed by this equation. Therefore, you can calculate the probability of the combined outcomes by multiplying the probability of the individual outcomes. So the probability that there will be at least one dominant allele for both genes is 3/4 multiplied by 3/4, or 9/16. The other probabilities are 3/16, 3/16, and 1/16. Note, this is the same ratio we obtained with the Punnett square. Eventually, we can dispense with diagrams and jump straight to calculations. For example, let's consider a 3-factor test cross with the same dominance characteristics as before. What is the probability that offspring from this cross will possess at least one recessive phenotype? We would be interested in offspring with one pair of recessive alleles, with 2 pairs of recessive alleles, or all 3 recessive alleles. For our first group, the probability calculation is 3/4 times 3/4 times 1/4, or 9 out of 64. The calculations for the other phenotypes are essentially the same. For the second group, the calculation is 3/4 times 1/4 times 1/4, or 3/64. And the last probability is 1 out of 64. To obtain the total probability, add all of the fractions together. This ends up being 37 out of 64, or about 58%. When dealing with the probability of Mendelian genetics, Punnett squares are an excellent tool to determine the possible outcomes of many different crosses, in addition to the heterozygous crosses we've examined today. You can then build upon these simple probabilities to calculate more complex outcomes.